2014, question number 13. A green fluorescent protein, GFP, encoding gene is fused to a gene encoding a specific protein for expression in the cells. So, what have they done? They have taken the gene for green fluorescent protein and they have fused it with the gene for the normal protein expression in the cell. So, they have done a fusion protein. They have made a fusion protein. What does a fusion protein means? That they will be having like if the transcription start site or the translation start line will be ahead of this, and they will be created as a single mRNA, okay, and then they will be translated into the protein. So, why are we using? What is the advantage of using GFP over staining cells with fluorescently labeled antibodies that bind to the target protein? So, you know that for detection of protein, we can normally use like antibodies, fluorescently tagged antibodies that bind to the protein. We can have a protein, okay, any like this protein is there, and then we have an antibody which binds to certain epitope of that protein, and with that antibody we can bind another secondary antibody will be there which will be tagged to a fluorescent probe, and we can detect this probe under the microscope or under western blotting conditions. Okay, this is the normal procedure we follow. Here they are saying staining the cells with fluorescently labeled antibodies. So so what happens during when we try to stain a cell? So what we do is we fix the cells, and then we fix the cell. Fixing the cell normally we do with methanol or paraformaldehyde, and then we so this is in fixed condition, but it is unpermeabilized. Okay, the cell is not permeabilized. You see that the antibodies cannot enter the cell cytoplasm because they are big, and the pores in the in the plasma membrane are not big enough so that the antibody can cross the cytoplasm. Now you give saponin or you give tritoxantrate. So both of these are detergent. Okay, so when we give saponin, it is a mild detergent, so only the plasma membrane is has these kind of pores. So the antibody can stain the proteins which are present in the cytoplasm. But when we give Triton X100, then you see a larger pores are getting created, right? Right, larger pores are getting created, and they are also present in the nuclear membrane. So you can also stain proteins which are present in the nuclear membrane. But in the case of saponin, you cannot. In unpermeabilized, you to definitely cannot. So what we do, we fix the cells and then we permeabilize the cells using a detergent, and then we stain with antibodies so that okay we give our desired antibody specific to the protein of interest. And what will happen? Then we give a secondary antibody which is fluorescently probed, and then we do any kind of assays. We can do flow cytometry, Western blotting, no, not Western blotting. You can do flow cytometry. You can do normal confocal imaging. So these are the things. So, but what happens is that fixation. When we say fixation, we kill the cells. Okay, fixation always kills the cell. So what happens is that when we use a fixative agent, normally we use methanol as a fixative agent or paraformaldehyde, anything. So fixation by methanol, which is an alcohol, it removes the hydrate cover. So okay, in of the cells, this is the hydration layer. So it removes that, causing the proteins to collapse and refold in this process, rendering them insoluble. Okay, methanol is merely the fastest penetrating, so we use that. So another fixative agent is aldehyde or paraformaldehyde that also causes pores. But any fixative agent, whichever you use, they cause death to the cells. So why are we talking about this? So if we see the options, So now we have created a protein, okay, which has a GFP tag. So we have a fusion protein. So whenever this protein will get expressed, it will be a fusion of GFP and the specific protein. So why are we using this process instead of the normal conventional antibody staining technique? So this for A, it bleaches less compared to the fluorescent probes. So why is it because the GFP bleaches less compared to the fluorescent probe? Or B, it allows imaging at higher resolution than the fluorescent probes? C, it provides more precise location of the anti or of the protein than the fluorescent probes? Or D, it allows tracking the location of the protein in the living cells. Okay, this is a point here. While staining usually requires fixation of the cells. So, which of these options is the correct one? First of all, the bleaching or the higher resolution imaging or precise location, all the 3 points can be achieved by normal fluorescently labeled antibodies. All of these have the similar kind of effect if we compare that activity with that of GFP. Now, in the last point, if you see that its fusion allows tracking the location of the protein in the living cells. So that is a point that is normally very difficult when we use the antibody method, because in order for the antibody to go inside the cell, we have to fix and permeabilize the cell, and that causes death of the cells. So you cannot track the live location of the antibody or the live location of the protein. So whenever you have to see something in a live cell or you do a live cell imaging, then you have to use this technique. And so this is the advantage of GFP over using fluorescently labeled antibodies.